cha. Topic: Intro to differential staining. Differential staining uses multiple reagents to distinguish between different types of bacteria or structures. Different from simple stains, which only use one dye and show size, shape, and arrangement. Gram stain. 1. What four things can a gram stain tell you? Cell shape, arrangement, gram reaction, positive or negative, relative cell wall thickness. 2. Why do gram-positive cells stain purple and gram-negative stain pink? The thick peptidoglycan of gram-positive traps crystal violet, keeping it purple. Gram-negatives have thin peptidoglycan and an outer membrane, so the crystal violet rinses out and they take up safranin, pink. 3. Cell wall differences. Gram-positive, thick peptidoglycan, no outer membrane. Gram-negative, thin peptidoglycan, has an outer membrane. 4. Gram stain steps and functions. 1. Crystal violet, primary stain, all cells purple. 2. Iodine, mordant, locks dye in gram-positive. 3. Alcohol, decolorizer, gram-negatives lose color. 4. Safranin, counterstain, stains gram-negatives pink. If steps are skipped, no iodine equals dye not fixed. No alcohol, both types stay purple. No safranin equals gram-negatives remain colorless. 5. Differential step, the alcohol decolorizer. It separates gram-positive from gram-negative. 6. Importance of controls. Having known gram-positive and gram-negative bacteria ensures your stain was done correctly. 1. Examples: Mycobacterium tuberculosis, Mycobacterium leprae. 2. What makes them acid-fast? High mycolic acid, waxy lipid in the cell wall resists normal stains. 3. Can you gram stain acid-fast bacteria? Not effectively. Waxy wall prevents stain uptake. 4. Stains and steps. 1. Carbofuchsin, primary stain, heat helps penetrate. 2. Acid alcohol, decolorizer. 3. Methylene blue, counterstain 5. Differential step. Acid alcohol, acid fast retains red, non-acid fast loses color. 6. Why do acid fast staining? It identifies pathogens like tuberculosis, which don't show up well on gram stain. 7. Final appearance. Acid fast, non-acid fast blue. Capsule staining. 1. What is a glycocalyx? A sugar coating around bacteria. 2. Capsule makeup and location. Made of polysaccharides or polypeptides found outside the cell wall. Functions. Prevents phagocytosis, helps bacteria stick to surfaces, protects from dehydration. 4. Colors after staining. Background dark, acidic stain. Cells stained, basic stain. Capsule, clear halo, does not absorb stain. 5. Type of stains. Background acid, e.g. Congo red. Cells, basic, e.g. crystal violet. Capsules remain unstained. 6. Why negative stain? Capsules are delicate and won't take up stain. Negative stain shows contrast. 7. Heat fix? No, heat can destroy the capsule. 8. Can gram stain detect capsules? No, it doesn't show capsules clearly. 9. Why differential? It distinguishes between capsule producers versus non-producers. 10. What four things can you tell? Capsule presence, shape, arrangement, relative size. Endospore staining. 1. What is an endospore? A dormant, highly resistant structure formed by some bacteria. 2. When are they made? In harsh conditions like starvation, heat, or dehydration. 3. Examples: Bacillus and Clostridium. 4. Why do endospore staining? To identify spore-forming pathogens. Why vasoresistant? Dipicolinic acid and a thick spore coat protect the spore. 6. Simple stain? No, the spore resists staining. 7. Stains used: malachite green equals primary stain spores, steam equals mordant, decolorizer equals decolorizer, stains vegetative cells. And if 8, final appearance. 8 spores equals green, vegetative cells equals pink. Differential step: the decolorizing with water separates spores from cells. 10. How to tell apart: spores equals round, oval, green, vegetative with pink rods. 24-hour versus 7-day bacillus. 24-hour equals mostly vegetative cells. 7-day equals more endospores formed. Well, why differential? Only some bacteria make spores, visible with this stain. 13. What can you tell? Spore presence, location, shape, bacterial identity, genus level. Flagella staining. 1. Function. Flagella help with movement. Not all bacteria have them. 2. Arrangements. Monotrichous, 1 flagellum, amphitrichous, 1 on each end. Lophotrichous, tuft at one end. Paratrichus, all over. 3. Why stain flagella? They're too thin to see without special stain. 4. Why is it differential? It shows which bacteria have or lack flagella and the type. 5. What 4 things can you tell? Presence, number, location, bacterial ID, to group. Group. 1. Where do they grow in thioglycolate broth? Obligate aerobes, top, obligate anaerobes bottom, facultative anaerobes, mostly top, some throughout, aerotolerant, evenly throughout, microaerophiles just below surface. 2. Capnophile, loves CO2, grows best in high CO2, low O2. 3. Why is oxygen toxic to some? It forms superoxide and hydrogen peroxide, which are toxic. 4. Enzymes needed, superoxide dismutase, SODs, catalase is most essential. 5. Which types have these enzymes? Obligate aerobes, facultative, aerotolerant equals have enzymes. Anaerobes don't. You'll know by where they grow. 6. Growth conditions in lab, obligate aerobe, incubator, obligate anaerobes, gas pack, capnophile, microaerophiles, candle jar, facultative, anywhere, best with oxygen. 7. Growth patterns, see where they grow in tube jar to determine type. 8. Anaerobic jar, removes oxygen using gas packs that create a hydrogen and CO2-rich environment, plus a catalyst to remove O2. Pathogenic cocci and media. 1. Pathogenic cocci, Staphylococcus, Streptococcus, Enterococcus, Neisseria. 2. Staphylococcus versus Staphylococci. Staphylococcus is a genus. Staphylococci will cluster-shaped cocci arrangement. 3. Gram reaction shape arrangement. Staphylococcus, gram-positive cocci clusters, gram-positive cocci chains, Enterococcus, gram-positive cocci, Neisseria, gram-negative diplococci. 4. Staph species, S. aureus, pathogenic, S. epidermidis, non-pathogenic. 5. Streptococcus classification, Lancefield, based on carbohydrate antigens. Hemolysis, beta equals clear, alpha equals green, gamma equals no change. 6. Selective versus differential media. Selective equals inhibits some, lets others grow. Differential equals shows visible differences. 7. Media types. PA, selective for gram alcohol. MSA, selective high salt, differential, mannitol fermentation equals yellow. Blood agar, differential hemolysis. BEA, selective and differential, bile and esculin hydrolysis. Coagulase test detects enzyme A. 8. Catalase test, substrate and peroxide, positive bubbling. Staph equals positive, strep equals negative. 9. Coagulase test, tests for clotting enzyme, S. aureus positive, S. epidermidis equals negative. 10. Media to tell staph species apart, MSA, coagulase test. 11. Media to tell staph versus strep, catalase test. 12. Media ingredient changes, remove inhibitor, not selective, remove substrate, not differential, remove indicator, can't detect difference, still selective. Let me know if you'd like this formatted into audio or flashcard.